great pleasure. It's a great pleasure to welcome Andy Lakoff to the reading group this week. We've got a number of articles, actually a lot of things that Andy asked us to read. And maybe we'll just start by diving into Unprepared. So you've shared with us chapter 5, a chapter that deals directly with gain of function research. And here we're learning about the history of H5N1 research in the US. And this group of researchers that were trying to publish their paper and running up against bureaucrats at the NIH that were saying, there's certain kinds of things we don't need to be publishing. And we haven't talked directly about gain of function research in this reading group yet. And the fact of the matter is that it does happen. There are people who do this kind of research. And, maybe if you could just kind of update us, from this book, the chapter that we read at least was focused on the US, what kinds of other gain of function research is out there? And then maybe we can kind of branch out and start talking about, some of the other readings that you offered up today.
Sure. Well, let me just first begin with thanks for the invitation. And I'm really happy to see a bunch of friends out there and people who I haven't met whose work I've read. So it's exciting to be here and have this conversation. And, one surprising upside of this pandemic has been the possibility of suddenly gathering at 7:30 a.m. in Australia and 9:30 p.m. in Central Europe. And here we're at 12:30. So this is really exciting for me, and I'd love to hear the reflections of various members of the group on this talk and on the material that I circulated, not just my own material, but the other stuff as well. I should say that the book as a whole is not about gain of function research or really focused on biosafety per se. It came out in 2017, and it was kind of a series of controversies around the possibility of a future outbreak of dangerous disease, whether that outbreak was human caused or of natural origin. And the book tracks what I call the dispositif, for an apparatus of preparedness as it emerges and consolidates in the late 1990s beginning in the 1990s and into the late teens. And obviously I didn't foresee this pandemic, and it's been a funny experience to have published this book a few years ago and sort of try to engage with reporters and others during the pandemic, because of course the title of the book, Unprepared, I think will immediately make people think I'm one of those folks who's been sort of sounding the alarm, saying, we're unprepared. We need to engage in more viral surveillance. We need to get our public health response systems in a better readiness mode. But of course, the book is not that. It's really more of a diagnosis of how we came to think we're unprepared and what preparedness means, what it means to do preparedness. I thought I would share this chapter with you guys for a couple of reasons. One is because, of course, there's ferrets are there and viruses, so there's various species along with humans involved in this chapter, whereas much of the book is actually more about expertise per se. But also because I think this topic is an interesting one to broach, and not one that I've taken it. That is the topic of the possible role of gain of function research in the inception of the current pandemic. And it's not one that I have a position on. Myself, it's not one that I've taken a really deep dive in, besides reading some of the work that I sent to you all to take a look at. But I think we're getting to a moment where the conversation, I'm guessing, will become more intensive around we'll sort of renew around the question of origins. And I think it's for critical analysts thinking about the stakes of figuring out the origins, the stakes of origin stories is an interesting and important one. And one reason I think is because we're getting to a kind of assessment phase, post hoc, even though we're in the midst of the pandemic still, we're also getting to a kind of post hoc assessment of failure phase in which various commissions of inquiry are getting put together. And the role of those commissions of inquiry is to diagnose why the response went so amiss in the United States, whereas it looked much better in places like South Korea or Australia. And therefore to take those diagnoses and use those to make prescriptions. Okay, so what do we need to do to prepare for the next pandemic coming down the road? And to me, the origin story, what comes out of the origin story question is going to be really interesting in that regard, because if you're one of the people from the world of One Health, who does viral forecasting and wants to gather to do ongoing molecular surveillance of animals that might have viruses with zoonotic potential, spillover potential, you might reap a lot of benefits from this assessment process. You might be able to get a lot of resources for your labs. But of course, if the origin story results in a story, in a evidence of an accidental lab leak from a biosafety level 4 laboratory, then the direction of future preparedness, the implications for that are quite different. So I'm guessing that this conversation, even though right now it's sort of peripheral, and it's, if any journalist writes a piece like the one in New York Magazine, they're in a lot of danger of being called a conspiracy theorist. I think it's possible that it will percolate up, but also in the US, the topic has been, as Baker mentioned, pretty taboo because of its association with Trumpism. And anti-China discourse from the State Department. But now that Trump is no longer in office, that might also make it more possible to open up the conversation in the U.S. So with that, why don't I pause and let you guys ask any questions you want.
And yeah, if people are joining for the first time, yeah, feel free to raise your hand, ask questions, or just unmute yourself. Zhush.
Yeah, mine is sort of a question comment, but I was really interested in sort of in the piece itself, it wasn't mentioned, but the militarized histories of gain of function research. So especially maybe taking it towards a different geographical location, because I know that one of the criticisms around the bioethical implications is that it's usually very US centric. So we've discussed US organizations, US bioethic frameworks, and I just wanted to shift maybe sort of the geographical lens to the Soviet Union, where gain of function research was so intertwined with militarized and weaponized viruses. So even under, I think, Gorbachev's weaponization-based GOFR, it was all about using viral vectors, not for scientific knowledge making per se, but for biosecurity and for very particularly militarized biosecurity. So I was wondering if we could engage with those questions of how do we reconcile this intensely militaristic history with the current bioethical questions around the research, and maybe how to negotiate these haunting specters of history around gain of function. That was sort of like my little invocation, but yeah.
Should I jump in? Yeah, I mean, I think that in general, the question of the relationship between biodefense and pandemic preparedness is, and they're integrally related. And I actually, you may know more than I do about the military history of gain of function research per se. And that's actually really interesting what you say about the Soviet history there. I guess what I would say is that the whole effort that this agency that I trace a little bit of in the chapter that you read, the NSABB, was founded in 2003, I believe, in the context of biodefense and biodefense funded research. So it wasn't explicitly about gain of function in the US, but it was certainly about the felt need to massively expand biodefense research and outside of the world of military research to the broader academic community, where high prestige, high profile researchers who were going to be able to do much more cutting edge genomic research were located. And so the funding of BSL laboratories was getting into motion. BSL 3 and 4 labs was getting into motion. And so that was when this sense of a need for some form of oversight emerged, that is around the demand for publication by academic researchers. Because of it, when it was purely in a military context, of course, the military scientists were expected not to publish their results in confidential studies in biodefense. But once it was extended out to the academic world, those academics funded by the NIH expected to be able to publish their work. And so then the question became, okay, so when is a publication dangerous? And that's where that was the sort of administrative regulatory space in which US gain of function circa 2005, 2006 came to be put into discussion.
No, that's really fascinating in terms of even how sort of different industry pressures sort of speak to one another and interact with one another. Of what happens when you take it into this other sphere where you have other priorities and other hierarchies of value, and also I guess when it comes to patenting and the intellectual community's relationship with patenting, also this newfound thrust of wanting to share and communally engage with knowledge, and the opposition towards the privatization or the classification of particular prototypes of patents as undemocratic or unscholarly in a way too. Interesting. But yeah, thank you for.
And if I can add one more point, since you raised the question of the sort of geographical dimensions of this, I think that, even though it's been cast as a sort of nationalist conversation about blaming Chinese science, what we're really looking at in this field of gain of function research now is a cosmopolitan form of science, often funded by places like the NIH in other places, in other nations like China. So it's an extensive cosmopolitan form of knowledge production that isn't nationally based, except when it becomes militarized, as you say.
I in particular was interested in your discussion of Asilomar. I've just finished a book on CRISPR, and one of the key moments is David Baltimore, the guy who sort of convened Asilomar and has been promoting scientific self-regulation for 30, 40 years now, getting up and admitting a moment of failure when Dr. He Jiankui, created the world's first CRISPR babies. And to me, this discourse within the sciences about self-governance and self-regulation sits easily within a neoliberal framework. And within the space of biotech and genetics in particular, it seems like the regulation has been taken over by the security agents, where the FBI is kind of policing the bounds of in biohacker spaces or elsewhere. It's not sort of a broader deliberative process where you have multidisciplinary panels coming together to decide what research is worthy of support or what should be quashed. The rhetoric around regulation gets securitized, and I guess maybe it's an invitation to comment sort of against the backdrop of that history of Asilomar, thinking about regulation in terms of security and the peculiarity of the American project as it sits with a neoliberal logic.
Okay, I'm gonna see whether an answer with respect to neoliberalism pops up, but not start with that, and talk a little bit about the legacy of Asilomar, which I think is super important in the story of gain of function research in the US, but now internationally. So Asilomar is typically much heralded as a model for scientific self-regulation, as most of you probably already know. This mid-1970s agreement that genetic engineering research, that there would be a way to allow scientists to follow their own interests and advances in doing genetic engineering and rely on them to, in some sense, police themselves through these local level biosafety communities. I mean, the real goal was to sort of allow the science to continue and to stave off public worries about the possible implications of genetic engineering. So I think public critics have often found the Asilomar agreement to be somewhat dubious. And then this is sort of before the entrance of security regimes. There was already a lot of criticism from STS scholars as well as other critics of the Asilomar model. But Asilomar really was sort of the available model when the gain of function and other kinds of newly controversial post-human genome project research came up. And the goal, as Fauci said in the quote that I had in that article, is to kind of enable scientific research to continue and to again be able to bracket off public concerns via this sort of risk analysis approach. And so that risk management approach goes back to the Asilomar model. But what happened as of the early 2000s is that defense and biodefense and biosecurity folks started to worry about, they would often call it the bio Unabomber model, but like the figure of the biological researcher who becomes a bioterrorist, and whether the Asilomar regime was actually adequate for regulating against that kind of possibility, or the possibility of groups of foreign scientists working with an enemy state to develop bioweapons. And that's when a sort of more securitized set of proposals have emerged around, and then of course the biohacking era that we're in today has sort of exacerbated that, and worries around synthetic biology. So I think right now you have at least two different conflicting models, one of which is sort of the self-regulation of scientific practice that comes out of Asilomar versus this more security and defense orientation that is state-based, and that comes out of anxiety that new developments in biotechnology render that, and the spread of those, the capacity. The other thing to say about Asilomar is that it assumed a fairly circumscribed community of high-level elite bioscientists, mainly in the US or maybe Western Europe, that would be able to kind of come to an agreement about what the right regulatory practices should be. And now we live in a totally different universe in which that is not what cutting-edge bioscience looks like.
Soraya, you've got your hand up.
Yeah, I also wanted to make those points about Asilomar, and it was not just about this being a model or the end actually of a way of regulating, because also for the life sciences, because it was not just that it was just the scientists deciding, but excluding the public. It was a clear way. I mean, also there were journalists in the meetings, but they were not allowed to publish before. Imagine this nowadays. They were not allowed to publish before the end of the procedures. And then they did. And then, it wasn't the end. It was actually the beginning of the public really becoming involved and worried. But also because it was a strong reduction about how to think about the risk that was involved, right? And limiting it actually to the health risk and excluding, for instance, environmental risk, evolutionary considerations, or ethical considerations. So it was a strong way of getting, of keeping it in the whole discussion in a particular way, and still they afterwards, people like Watson said it was what was it? He said a folly or something. It was a folly to even try to do that. We should never have done this. Even if it was in this way, sort of controlled and yeah. So I think so. I wanted to say something about this Asilomar thing, but I have another question, which is much more basic, and it's actually maybe it's one for you, even because you said in your invitation thing here to explore the dynamics of the gain of function experimentation and the re-emergent discussion of the lab leak hypothesis. So my question is that really re-emerging, or are we making it to re-emerge just now? Because I thought this was actually somehow close, that some people had looked that this could not have been a lab thing. But it reminds me of the conversations around AIDS, right? Because I don't know if you remember, some of you may remember, that there was the same conversation, right? That this maybe have been escaped from the lab. And I remember the discussions at the time that said, even if it hadn't happened like that, and it seems very clear that it hasn't happened like that, just thinking that it could be possibly have happened that way also already tells us, right? So we don't need to know if it really happened that way, but just the possibility that it could have happened that way actually should already, it's already important, and we should think about the consequences of that. But the basic question is it really re-emerging, or are we just resurrecting it in this seminar here?
Well, for starters, to respond, Soraya, Rachel wrote that blurb, so I didn't write the blurb, but and she's teaching right now, so she can't sort of contextualize the blurb. But I should say I do have a strong opinion about the lab leak hypothesis, and in print I've already said that I think the evidence is flimsy, and we can go through the Baker piece and kind of push and poke at some of the particular things that Baker is claiming about the lab emergence. But one thing that I observed both from the Baker piece and the PNAS piece that you asked us to read, Andy, is that, so Baker says, both sides, and here he's talking about folks who think that, there was this kind of emerged in animals and then infected people, and the other side is the lab emergent hypothesis. So Baker says both sides agree for the most part that the spillover event that began the human outbreak probably happened only once or a few times quite recently, and not many times over a long period. And actually, that's not quite right. A similar point is suggested in the PNAS piece. And the fact that Zhush is with us here today, experiencing basically no symptoms, points to the fact that this disease could have been circulating for a long time before. Bye, I just got Zoom bombed by a 5-year-old. Hello, goodbye. So SARS-CoV-2 could have been circulating amongst humans, or a relative of SARS-CoV-2 could have been circulating amongst humans for a long, long time without sort of getting picked up and noticed. I'll turn it back to Andy, but I did want to say, part of me reads this with the medical anthropology literature that's about, social causes of disease. There's a long literature that, like in New Guinea or Africa, where witchcraft allegations are associated with somebody getting sick. There's often a social explanation, and I guess part of the bias that I'm bringing to the conversation is the bias that humans aren't exceptional, and that for me, one of the things that the pandemic illustrates is kind of shared biology that humans have with other kinds of animals. And I guess my reading of the pandemic is an interruption of the modern project. This idea that sort of we can know and understand the diversity of viral species that are relevant to us. I think if anything, it's revealing how little research was being done on coronaviruses, how little we understood about the potential of these kinds of viruses to make us sick. But I'll let you respond too, Andy.
Actually, Frederic, you had your hand up a little bit ways ago. Would you want to respond to this issue about that Soraya raised around whether the debate is re-emerging, or just what the significance of the origin question might be?
I wanted to raise the discussion on another issue, so maybe I'll let you answer on the re-emergence of the debate on gain of function.
Yeah, well, I guess just initially on this question of is it actually re-emerging, the question that Rachel posed in absentia for us. I'm not sure. I think it's pretty marginal. I think there is a sense of a scientific, at least a semi-consensus, that the most likely explanation is a spillover event. But I'm detecting a sort of, I detected, when I got this invitation to speak with you all, as sort of something of a re-emergence of the discussion, not only in that New York Magazine piece, but also this piece from Boston Review, interviewing a scientist named Alina Chen from the Broad Institute, and some of the discussions by biosecurity experts like Leitenberg and Philippa Lensos, and then in a few other media contexts, like a Wired piece that came out by Roger Pielka Jr. So I'm seeing it pop up. I don't think that it's massively re-emerging. I guess I think, Soraya, your last point about is the very fact that it's plausible something that should be an object of concern, which I think is also the point that Baker and Pielka are raising, which is, I think that's true, and that if one of the prescriptions of the post-pandemic action plans is we need to invest a lot more money in basic bench virology research, including lots and lots of gain of function research, we might want to pause and say, okay, wait, there have been lab leaks, not necessarily with the coronavirus, but over the last 5-10 years, there have been incidents of lab leaks in biosafety level 4 facilities. Do we want to be funding in the US and or globally a lot more of this kind of research? And if it's plausible that it can spark a pandemic. So I think that's the kind of question that one might pose, whether or not one has a position, whatever one's position is about the origins of COVID.
Fred, do you
Can I
want to
jump
ask your
in?
question?
Yeah. But I agree that the debate on the origin of pandemic viruses is in some way repetitive, and it's great that Andy raises the question in such a way that we see the consequences of the debate on the technologies of viral research, such as gain of function, lab research, and I'm interested also in the debate about the cold chain that allows to keep viruses and to trace viruses back. So raising the question about the origins in social sciences, not in terms of accusation, but rather in terms of technological consequences is great. But I wanted to raise the discussion on another issue, which is the timing of the discussion of the origins. So why the debate on the origins of COVID-19 emerges 1 year after the first outbreak, at a moment when vaccines are available, when a lot of Asian countries seem to control the pandemic, while a lot of European and American countries seem not to control. And I'm engaged now in a debate about zero COVID, which is very much debated in Europe between those who say we should do like Asian countries and stop COVID when it enters our borders, and others who say no, we should live with the virus and use the vaccines to get a mix of natural and artificial immunity. So I mean, this coincidence between the debate on the origins and the debate about zero COVID looks like we want to go back to a time when COVID-19 doesn't exist, and we look at Asian countries as those who are closer to the time of origin in some way. So, I mean, do you have ideas about this question of the timing of the debate?
I mean, I guess it gets back to the question of is there even a debate? Which, you know, I'm not certain there is. I think I'm curious as to whether there is, and especially because we have an international audience, whether the debate if there's a debate, whether it looks different in the US than in other countries. I think it's possible that if a debate emerges, it would be more open now than it would have been 6 months ago, because in the US, because during the Trump administration, scientists with progressive leanings were very hesitant to look like they might be endorsing a view that would blame China, just by even opening the question. I also think that the debate, if there's a debate again, it may be opening up now just in terms of timing, because WHO just launched this investigation. There's a team of researchers that went to China to look into the origins. Now, interestingly, one of the leaders of that team is this guy, Peter do you say Daszak? I'm not sure how to pronounce his name. Who's a major figure in the One Health movement and had fostered the collaboration between NIH and the laboratory in Wuhan that is in question. So there's an issue around conflict of interest if he's the one leading this investigation, and is also one of the ones who in February made a major effort to shut down discussion around around the possibility of a lab leak as the origin. And he's also somebody who would benefit from a lot more infusion of funding into this study of animal human transmission and viral evolution as a part of the pandemic preparedness apparatus. So that's a sort of a scattered answer, but I think that maybe one reason why a debate may reemerge has to do with this sort of moment of assessment that we may be entering into of, okay, so, now that we understand a little bit more about the disease and maybe are getting closer to managing it via vaccination, can we step back and see, why it was that some countries responded well and others badly? What do we need to do in the US and elsewhere to respond better to the next one? And I think it's this next one. I mean, that was one of the things that sort of led me to this book, was this sort of discourse of the next one. There's always a next one down, the next emergency down the road. And you're really seeing that very commonly in discussions of COVID. Okay, well, COVID was bad, but that wasn't the really bad scenario. The really bad scenario is the one that, has a much higher case fatality rate and hits young people. And that one's coming down the pike. And that's the one we need to prepare for. And so it's, in a sense, this actual event is being treated the way that simulation exercises are being treated, and that they expose sites of failure and tell us, where we need to improve. And I think insofar as critical analysts can enter into the discussion, we might say, well, wait a minute, is that form of preparedness really the thing we need to do next?
So Shush, you have your hand up, and then I do want to get into the weeds a little bit and interrogate some of the evidentiary claims that Baker makes.
I'll be very, very quick. No, I think my in terms of that responsiveness to the virus itself as a conceptual assemblage, I think it's also interesting to see the effect of observing a virus that is, that holds so many variations, and and the impact of that on the population. Because as the virus has been shifting and we're getting to know different variations, and there's been the Brazilian, there's been the South African, the British variant, it does sort of bring this conceptual premise that this viral measure is not static, and I wonder what that does in a conceptual terrain to understand these viruses as constantly shifting and evolving things, and whether that has actually led us from this initial paranoia, which marked a lot of the COVID discourse at the start of the virus, of the potential lab leaks, of the quite xenophobic and racist undertones of the Wuhan leak. And then we sort of entered this domain where we were starting to get to know more about the zoonotic carriers and the various potential vectors of the virus. So we were seeing variants in species, and now we're also seeing variants in viral formation and configuration. So there's this sort of plurality that's entering the discourse, mainly because there's been so much knowledge about the variants, the sheer diversity of one virus itself. And I wonder if that is leading us away from more static or paranoid readings. But that's sort of just a speculative thought, I guess. But yeah. Evan.
Go ahead, Andrew, if you want to respond.
No, yeah, let's continue.
Okay, cool. Well, actually, that kind of builds into some of the things I was about to say, kind of in a conceptual register. So early on, we read Stefan's paper on the trees and seeds of information, and I wrote something that kind of in a Foucauldian idiom refused the search for the origin, but instead was trying to think about conditions of possibility for emergence, which are different questions, right? And going back to that bit from the Baker piece that I read about, claiming that there's a consensus about a single origin. In fact, there's not. And what we find when we look at these things are, snapshots of genetic diversity that, as Josh was just saying, is very dynamic. With, Deleuze teaches us that, we make rhizomes with our virus. These genetic components are moving amongst kingdoms sometimes, as well as amongst these different variants or strains. So the coronaviruses are different from influenza viruses. They don't have chromosomes, so they can't reassort, but they can recombine. And that's a possibility, that there was some kind of benign coronavirus circulating in people, or in people in multiple animal species, and there was a recombination event. So one of the bits of information that this Baker piece goes over fairly quickly, it's engaging with Vincent Racaniello. He's the host of This Week in Virology. He's a professor at Columbia. He authored the textbook Principles in Virology. So Racaniello and many others have pointed out that this has not been seen before. So looking at RATG13 and the SARS-CoV-2 strain, you see approximately 1,200 point mutations that weren't there, that are there in SARS-CoV-2, but that weren't there in RATG13. So just a couple of weeks ago, Racaniello and his friends on the TWIV podcast, This Week in Virology, we're talking about who would do that research. So you've got this coronavirus sequence stored in your database, RATG, and to induce 1,200 point mutations, that's a lot of money, that's a lot of time. And why? You could think about you know, some of the experiments that you were describing are about, you know, passing it through, you know, various hosts, but to induce that many point mutations would take a lot of work and a lot of time. And so that's one of the main points from that kind of consensus statement, that is looking to evidence that this was not manufactured in a lab. The piece by Baker also mentions a paper by Christian Andersen. And I think Christian was in the third meeting that we had of this reading group discussing his paper in Nature. That was a careful accounting of you know, the genomic sequence of SARS-CoV-2 and doing some of the bioinformatics work to look at the other viral strains that it was related to. So that paper is titled Proximal Origin of SARS-CoV-2. And one of Christian's points in that paper is that this particular spike had not been seen before, that the kinds of binding motifs, the dynamics of molecular function just hadn't been seen. And to create that de novo, to sort of invent that, someone kind of sitting by themselves thinking like, oh, I want to design a spike that works really well, I'm going to tweak this, this, and this. He said that was really unlikely, in the sense that what synthetic biologists are really good at is, you know, if you go to iGEM or any number of these other synthetic biology spaces, they're good at mixing and matching parts. So parts that have been, you know, established to have a particular function. But since this particular spike has not been described before and isn't cataloged in any databases, Christian's group, you know, just in addition to looking at how distant it was from the known sequences, yeah, he basically said, like, how would you invent this? It would be a really big endeavor, a really resource intensive endeavor to invent a spike like this. And I think maybe one other broad point that a number of visitors have shared with us, including Alexander Gorbalenia, the viral taxonomist that gave SARS-CoV-2 its official taxonomic name, is that this clade, this group of organisms, is massively undersampled. So there has been some focused sampling in bats, you know, but even bat coronavirus diversity is not very well understood. So you have these groups in Wuhan that have been studying bat coronaviruses, but kind of the specter of the rogue scientist in this paper, and I'm going to actually have to cue back. Let's see, do I still have this tab open? Ralph Barek, who's in the Department of Epidemiology, and also working on cancer at UNC Chapel Hill. He's painted in this Baker piece as a bat coronavirus expert, when in fact he's not. He is a coronavirus expert, but he hasn't been going out there and doing the sampling work like some of the colleagues in Wuhan have, and actually like some colleagues here in Australia. So it turns out that Geelong, near my office here in Melbourne, was probably the definitive center of coronavirus research before the Australian government, bat coronavirus research, before the Australian government shut it down about 4 years ago, citing like budgetary concerns. So some of the people here from the Geelong lab were involved in setting up the Wuhan lab. And yeah, to me, I guess I see less a story of sloppy lab practices or a story of really intensive investigation of this, you know, novel gain of function research. But yeah, a story of fragments of knowledge, and a story of, yeah, you know, we don't have a good understanding of, you know, the histories of biology in this case, to use a phrase from Hannah Landecker. You know, we don't have good archives going back. And I think for me, that's going to be some of the interesting work going forward, you know, as we find archived sewage samples, as we find archived blood sera, you know, can we start to study coronavirus diversity of even the common cold, which is really understudied. So that's kind of my response to the Baker piece, and I'd love to hear your thoughts, Andy, about you know, how you came across this, and your thoughts on, you know, sharing it with the group.
Yeah, and I mean, again, I think I tried to make clear in my earlier comments that I didn't share the piece because I want us to make a judgment about whether it originated in the lab or in in bats or in some transition species. I think it's provocative for me because in my study of the assemblage of pandemic preparedness, this discussion around origins has high stakes, and I think we're at a moment, as I said earlier, that there's going to be a real heightening and an intensification of that apparatus of preparedness, and I think we should be attentive to what that ends up looking like in terms of security regimes, in terms of public resources into public health, and in terms of basic research in fields like virology and epidemiology. So that's sort of my stakes here, without knowing the details that about the question of which bat virus might have mutated and how. I mean, I'll say and I'll call myself a layperson in this respect in comparison, and I'll dub you an expert even, but I will say that one piece of not just the Baker article, but some of the other sort of speculative pieces, I've been sort of curious to know how people respond to this mystery of the distance between the caves in South China and Wuhan. So we got 1,000 miles, right? And so and Wuhan is where the disease ostensibly emerges, or at least is detected. Even you're saying, well, there's lots of asymptomatic transmission, so there's no reason why actually Wuhan is the place where it was first circulating among humans. But I think it's it's an intriguing question why Wuhan is the spot where the disease seems to have made itself felt in the world. Yes, and it's the only place where they're doing BSL level 4 coronavirus research in China.
Yeah, I guess for me it's kind of the reverse, and I'd actually love to hear Wim's take on all of this too. So Wim's someone who works in the field and was the expert who identified the SARS-CoV-2 in mink populations. But yeah, for me, it's always been a question about where the critical coronavirus expertise, the zoonotic coronavirus expertise, was concentrated. And from what I gather, Wuhan is one of the best places in the world where you had a concentration of experts who were able to identify, recognize, and sequence this emergent pathogen. So Wim, I would love to hear your thoughts about the concentration of expertise at Wuhan and all the things we've been talking about.
Yes, thank you. Yes, I think it's very intriguing, and what we also saw in the mink outbreaks is that we see higher mutation rates in minks, and from what we observed there is that in the later phase of the outbreaks, we see that the virus becomes more virulent in the mink population. And we also see we're still working on all those information from the farms. We have called 70 farms, and we have obtained a lot of sequences. So we're still working on that. But we more and more have the impression that during the outbreak, during the circulation in the farm, in the high number of animals, the virus becomes more virulent. And of course, this is difficult to observe also in the human population because there's a lot of symptomless infections. So there's a lot of circulation that you don't even observe. And in the minks, that was the same. But at a certain point, the outbreak is detected, and at that point, it seems that the virus has become more virulent. That's more or less also what we see in the human population, because when it circulates longer in a certain area, we get these new variants, like the British variant, the South African variant, that seem to be hot spots of circulation of the virus. So there's a lot of mutations going on, and we have the same impression in the mink. And we see higher mutation rates in the minks, and that's probably due to the fact that these animals are kept close to each other in high numbers. So you have a lot of virus transmission, and there is more. It's it's more likely for the virus to have mutations that make the virus more virulent or easier to transmissible. And what I find also very intriguing about coronavirus, maybe the other people in the group have some opinion about that, is 20 years ago, coronaviruses were not even known to be zoonotic viruses. They were always, they were seen as viruses that were not zoonotic actually. And now we had 3 occasions. We had the MERS, we had SARS-1, SARS-2, 3 different viruses that, SARS-1 and 2 are related, of course, but now suddenly we have 2 of those viruses which are actually very zoonotic. And also the other fact that we also have the impression that coronavirus seems to be quite on the move, because also PDV, a pig coronavirus, has been emerging drastically over the last few years. So I find that intriguing, because since we are already talking about 4 different coronaviruses, and the coronaviruses seem to be emerging for some reason, and it's hard to explain why.
Thanks, Wim. And there's an interesting chat between Francisco and Stefan in the message chat about molecular clocks, phylogenetic evidence. Do either of you want to jump in and ask that?
Well, I'll let Stefan go first.
I mean, it's more of a set of questions that I have than anything else. I mean, I'm fascinated by obviously by the language of origins, and having been taught as a good Harawavian to be, if not skeptical of origins, to realize that origins are claims, and that there's an infrastructure that needs to be held steady in order for an origin to be secured. I wonder what those infrastructures are here. So just like the super elementary question, is there kind of like a foolproof forensic gain of function profile where you suddenly realize Darth Vader is your father, right? That like the kinship reveal, right? Is there something in the sequence that can do that, right? And I was just reading the paper that was published in Science last week, the analysis of the SARS-CoV-2 in the Boston Biogen conference, in which they say, well, we got all these samples from all around Boston Medical, and then we sort of, made some assumptions about mutation rates, and we had a pretty good idea of what kind of time frame we're working in order to figure stuff out. So it's not necessarily a molecular clock, but that's a molecular time frame. So I'm just curious about maybe Wim can help us out here with a sense of how far back to point 0 could you possibly get without making a whole set of assumptions about what institutions and ecologies are in play, and how do you know when you've crossed the threshold, to use the 1990s language, from nature to culture, right? Like, how can you see the signs of intelligent design?
Yeah, well, actually, this is something we're working on. We are at the moment we're gathering all the sequences of the mink viruses, and we are calculating. We are doing this molecular clock calculation. So there's more information to come about that. But I can't say much more than that mutations in mink seem to be, well, let's say 2.5 a factor 2.5 higher than in humans, but we're calculating that, we're working on that. So there's more information to come, and it's very intriguing. I agree.
Frederic,
go ahead, Andy.
Can I call on Frederic to talk a little bit about the cold chain and the role of freezers and all of this, and kind of how we think about how the cold chain has made possible certain kinds of attempts to track to retrospectively track the evolution of viruses from an origin?
Well, this is an information I got from another group on the origin of COVID-19, but it's basically some French and Belgians virologists doing molecular clock, and one of the hypotheses is that there's what they call a frozen evolution between the virus that was found in this cave in Mojiang and the one that was detected in Wuhan. So if you trace the branches, then there's a long branch which would indicate that the virus has been kept in the freezer, And so that's very interesting to compare it with other debates about maintenance of the cold chain in different occurrence of the pandemic.
So does that, I mean, how does that relate to what Aben was saying about the 1200 mutation events between the bat cave, the 2013 virus and SARS-CoV-2?
And I should point out, actually, one of the papers that Wim cited in his piece that he shared used a molecular clock to date the point of divergence to like 1981 is when the rat tag bat coronavirus and SARS-CoV-2 are thought to have a common ancestor, just working with the assumptions of the molecular clock. But go ahead, Frederic.
Well, it seems that you can trace the germs of the virus, but you can also trace the duration of the mutation. So when the mutations are pretty fast, it means that it spreads in nature, and when the mutations are pretty slow, I mean, it could indicate that it's been kept in the freezer. And so they compared that to, there was a blue tongue outbreak in Europe 4 or 5 years ago, and they could show also this phenomenon of frozen evolution. It's just an interesting expression to think about.
Shush.
And I do wonder about how origin stories sort of impose this linearity to the actual framework of a virus. Because I know that with, for example, Bonnie's recent paper on the evolution origins of SARS, there's equal weight given to sort of reservoir species. And how does sort of the importance of reservoir species come into the conversation alongside the origin species? So if we do ascribe this originary route to one species or one location or one event, how do we hold just as much space for the reservoir species or sort of potential intermediary species? And I think that's interesting in terms of the narrative enunciation or the narrative sort of onus that we place on certain species in this debate, especially of how do we recognize these alternative species that may have just as much that may contribute just as much, but sort of don't belong to that linear thread. So I do wonder about that, especially when we talk about genetic proximity.
Wim, I was wondering if you could say a little bit more, and we're kind of running out of time here, but just how molecular clocks work. It seemed like part of the discussion that was had just then was saying that time can speed up and slow down depending on kind of the environment that the virus finds itself in. So is your research suggesting that mutation rates happen faster in particular hosts, or was I mishearing?
Actually, I'm not a specialist in this, but we are simply looking in what others have done in molecular clock analysis and try to use that for the sequences we find in the minks also. So that's what we're doing at the moment. And I think it's an interesting way of looking at the transmissions in the populations. And that's what we're doing at the moment. And that's why we hope we can get some information to sort of discuss this again. We have a lot of questions about also not only within the populations, the development of mutations, but also we have a lot of questions how the virus transmits between farms. And that's also why we are looking very closely at all the different mutations we see. Some develop within the farm, and in some way they are transmitted to another farm. And we are trying to find that connections also. And so we're also doing distance calculations and modeling the different substrains also. So we're looking also in diversity, also looking at diversities of strains of the sequences in the farms to see whether we can find explanations for all the transmissions. And I think that could also be very helpful for what we see in the human population, but because it's probably not so very different. They're different species, but the development of the virus or the changes you see in the virus are probably not very different in the human population. So I hope we can get some explanation for that also.
Frederic, do you want to chime in there with what you're saying there, or Emma? Oh, you're muted.
What I saw on my Lakoff analysis for influenza viruses was based on the hypothesis that mutations were natural. So precisely, you could trace jumps between species, but there was not the hypothesis that there would be an artificial intervention, or except if there was a vaccine, you could identify the vaccine, the effect of massive vaccination. But here, I mean, this hypothesis of frozen evolution is a way to indicate a historical accident within molecular clock.
And maybe just riffing on some of the things that are being discussed. I mean, you could do targeted mutagenesis at a whole bunch of different sites at once. So you know, my book about CRISPR is all about how to induce mutations in DNA. And what you do is you come up with a guide RNA that's specific, usually when you're trying to change a particular gene sequence. But Adam Zaretsky, a bioartist who loves to sort of play with randomness and nonsense, working not with CRISPR, but with zinc fingers, another similar endonuclease that does targeted mutagenesis. He designed a guide RNA that was non-specific and that produced targeted mutagenesis at thousands of sites all at once. So, you know, you could do that research. So, you know, here maybe I'm sort of like contradicting some of the earlier stuff I was saying, or not contradicting, but just sort of pointing towards, like, if you were trying to induce mutagenesis within a genome, it's possible to design those non-specific guide RNAs. So yeah, maybe I'll go back to Emma.
Hi everyone, it's lovely to see you. I haven't been in this group for a while, just done pickup, and I've just been listening to the conversation through my morning. So I don't have a lot to say. I just want to just kind of commented, just bringing together what Eben was saying before about how do you tell nature from culture, and you know, the clock as a way of doing that, but also the capital and the flow of capital. I just thought that was interesting when Evan was saying, you know, would all the 1,200 mutations would be too expensive, so it's going to be natural. So just another way of accounting for nature and culture.
Brilliant summing up. Andy, do you want to have the last word here? And I must say, like, we've pushed and poked more at a piece, I think, today than we have before in the reading group. So thank you for offering up yourself as a sort of a pushing and poking, not quite a punching bag, but, like, you know, we had a lively conversation today, and I think it was great. But do you want to have the final word here?
Well, first, I learned a lot, and thank you for pulling this together. And I think that the questions that were posed are all really worth thinking about harder. I guess the one thing, just to bring it back to the piece that I centered around, and some of the points that you made, even, is that there's a kind of knowledge infrastructure issue at play here, which is, you know, how much do we know, and should we try to know about coronavirus and other kinds of viral mutations in the wild, in mink farms, in labs, et cetera. And that, you know, one of the major premises of the type of research I was discussing, gain of function research, is that you can link lab-based research on virology transmission to viral surveillance in the wild, and that's the premise of One Health, and that's why you would, NIH would in principle be funding this gain of function research, that we can, so that we could find, what makes a virus more easily transmissible about humans, and then have a massive surveillance apparatus that is detecting the emergence of that in the wild, and thereby, and then containing it before it gets to humans. And so there's a whole premise about the building up of a vast knowledge infrastructure about viral transmission in the wild that I think is what's going to ramp up post-COVID, or even before post-COVID. And that's part of what I'm pointing out, is this funny causal linkage, and it's a sort of funny epistemological claim that actually by doing this gain of function research, we could actually learn what to look for in the wild. So I think I just want to sort of underline that linkage that's being made by the field itself, and that's kind of my intervention there in that piece. So, but thanks again, and I hope I'm able to join you guys more in the future and hear from the next round of guests.
Yeah, please come back, Andy. We meet every Monday, yeah, I think your work is so critically important right now, and I think all of this sort of speaks to the need for critical genealogists, STS scholars, ethnographers to be in these sites that have become highly polarized and politicized, and those of us who are just trying to ask some of these basic questions. If the WHO is watching our YouTube channel, let us in too. But yeah, thanks again, Andy, and really great to see everybody, and see you next week.
Thanks, everyone.
Thank you. Bye.